In this topic, we're going to discuss the structure and function of phloem. So what is phloem and what is the structure of phloem? So we're going to look at the sieve tube elements and the companion cells. And then we're going to link the phloem structure to its function. So what is phloem? Well, this is the principal food collecting tissue in vascular plants. It carries organic material, for example, sugars and amino acids, from the leaves or storage organs to other parts of the plant. So it's actually a tissue made of sieve tube elements, as you can see on the left there, and the adjacent companion cells. You've also got parenchyma cells that act as packing tissue between other cells. So take a moment to have a look at the structure of phloem. Notice the difference between the sieve tube element on the left and the companion cell on the right. So sieve elements are elongated structures that are joined end to end to form long tubes. The cells are living and they retain a thin layer of cytoplasm within their cell surface membrane, and this lies against the cellulose cell wall, so we say that it's peripheral. Within the cytoplasm you can see mitochondria and a modified form of endoplasmic reticulum. However, they don't have nuclei. So they've got very few organelles, and the nuclei and the other structures are broken down in order to make the sieve tubes more hollow and so reduce resistance to the flow of liquid in them. So they've got no ribosomes and no tonoplast. They do have phloem proteins though. The end walls, as you can see on the left there, are perforated by large pores. So these perforated end walls are called sieve plates. You can also see plasmodesmata between the sieve element and the companion cells. Now, what do you notice that is different about the companion cell? Well, the companion cells are always associated with sieve elements, and both come from the same cell division. So companion cells are actually modified parenchyma cells. Now, if you notice, the sieve elements lack the structures, for example, nuclei, ribosomes, and Golgi apparatus, so they are unable to carry out the metabolic processes essential for their survival. Now, the companion cell are metabolically active because they've got these different organelles. So they've got a dense cytoplasm and a small vacuole, and they've got many organelles that assist with the metabolic activities. They've also got plasmodesmata, as you can see, between the two cells that link the two types of cells. Now these two cells, the companion cell and the sieve element, are so closely related that if one dies, the other cell dies. So here you can see a beautiful image of the sieve element next to the companion cell. So notice how the dense cytoplasm is in the companion cell. And also have a look at the large number of mitochondria in the companion cell. Also note the cytoplasm is around the edge of the sieve tube member. So we say that it's peripheral. The center of the sieve tube member, or the sieve tube element, is not a vacuole. It's actually an empty lumen through which the cell sap moves. The sap moves. So both these cells are connected by plasmodesmata. So here's another image of sieve tube elements. So you can see that they're elongated cells with perforated end walls. These are enlarged plasmodesmata and they form sieve plates. The nuclei have degenerated and the cytoplasm forms a thin peripheral layer. So the cytoplasm is actually continuous through the sieve plates, and notice how there's no vacuole. In this picture you can actually see the phloem parenchyma between the cells. It's actually quite difficult to see sieve plates in cross section. So this is an electron micrograph showing the large pits. There is a substance called callose around the sieve plates. This may help block off channels when damage occurs. So if you were looking under a microscope, you would notice the xylem, which stains red in the middle there. And then you can identify the phloem. So look for large sieve tube members and then the smaller companion cells next door to them.
Types of phloem. Sieve tube elements. Sieve tubes are tube-like structures composed of elongated cells arranged in longitudinal series and associated with companion cells. Their walls are thin and made of cellulose. In a mature sieve tube, the nucleus is absent, but peripheral cytoplasm as well as large vacuole is present. The main functions of sieve tubes is the transport of prepared food materials from leaves to the storage organs in the downward direction and then to growing regions in the upward direction. Companion cells. Companion cells are specialized parenchyma cells characterized by dense cytoplasm and conspicuous nucleus, closely associated with adjacent sieve tube element in the phloem of angiosperms. Phloem parenchyma. Phloem parenchyma cells are living parenchymatous cells which may be cylindrical, subspherical or polyhedral in shape. The cells have dense cytoplasm and nucleus and cell wall is composed of cellulose. Phloem fibers. Phloem fibers are much elongated, rarely branched and have pointed, needle-like apices. Their cell wall is quick-thick with simple bordered pits. At maturity, these fibers lose their protoplast and become dead.
Phloem, and we're going to differentiate between the two of them in terms of their structural differences and their functional differences. So let's begin with the xylem. The xylem consists of the transport vessels conducting water and minerals from the roots to the leaves. Now remember, these water and the minerals are going to be coming from the soil, so they need to get into the roots and be transported upwards to the leaves where photosynthesis can take place. So just recall that photosynthesis requires water as a raw material. So that is how the water gets into the plant. So these xylem vessels are very important in transporting that water from the roots upwards to the leaves for photosynthesis. Now the key points to remember about the structure of these xylem vessels. First of all, they're made up of dead tissue. So the reason that the tissue is dead, meaning that the cells are not living, this prevents any chemical reactions going on in the cells. Water can react with different substances in the cells to carry out different reactions, so we definitely don't want that happening. The whole purpose of the xylem vessel is strictly for conducting the water and the minerals upwards. So you don't want the water stopping and reacting with anything in the cells, so that is the reason why the tissue is dead. Now the second point to note is that the tubes are long, thin and hollow with no end walls. So this facilitates a smooth upward movement of the water. So as you can see, the third point says the flow of materials is upwards only, meaning that is unidirectional. So it's going from the roots to the leaves. So this long, thin, hollow tube is going to facilitate that movement, and the end walls is going to prevent any obstruction of the water flow. So the flow of water is assisted by the capillarity forces. So that is the forces between the water molecules and then the water molecules and the xylem walls. So we need to keep that water and the minerals going upwards. So think of the suction movement of water going through a straw. So these xylem vessels act like a straw for the plant. So the final point, the walls of these xylem vessels are lignified. So they have this substance which is usually found in wood. So you know wood is a very hard substance. So the lignified walls give the strength and the support to the xylem. So that gives it that strength, makes it strong, and helps to keep the xylem upright. Alright, so that is the xylem. Let's look at the phloem. Now the phloem consists of the transport vessels conducting organic material. And by organic material I mean mostly sugars. And these sugars would be sucrose. So this material would be going from the leaves generally to the roots of the plant. So just remember, in photosynthesis, glucose is manufactured. So that glucose is then converted into the sucrose, which needs to be transported from the leaves to the roots. So the key points to remember about the phloem is that it's made up of living tissue, unlike the xylem. So the living tissue is in the phloem, dead tissue in the xylem. And this living tissue consists of companion cells and sieve tube elements. Now, the companion cells are the cells that are going to be controlling the functions of both cells. So they consist of the nucleus, so they control all the activities going on in both the companion cell and the sieve tube element, which is responsible for the transport of the sugars. And you would notice that we have these perforations, or the end walls, which are broken down to form these sieve plates. And then you can see that the flow of materials is moving in both directions, upwards and downwards. So it's bidirectional movement. Alright, let's look at both of the vessels together. So you can see how they work together to transport materials. So the xylem is responsible for transporting the water along with the minerals, and the transpiration stream would help with that. So the water and the minerals are going upwards, while in the phloem you can see that the translocation of sucrose, so that's the movement of the sucrose from where they are made in the source, that is the leaves, to where they need to be used in the roots and other parts of the plant, so that is the sink. So that is the translocation of sucrose in the phloem. Alright, so you should be good now with the structural and functional differences of the xylem and the phloem. So hope this was a great help to you.